Welcome. Let's look at GenBank, which is a public gene database maintained by NCBI. In the address bar, we have NCBI National Center for Biotechnology Information .NLM National Library of Medicine .NIH National Institute of Health .gov. Let's retrieve the sequence for FLT-1, which is FMS, like tyrosine kinase 1, which plays a role in preeclampsia. FLT-1 encodes VEGFR1, which stands for vascular endothelial growth factor receptor 1. Is a common growth factor that we study. FLT1 comes in two forms. A membrane-bound FLT1 found on endothelial cells and regulates blood vessel formation. And a soluble FLT1, which, guess what, is SFLT1, which is a splice variant that binds VEGF and PLGF. Again, VEGF, vascular endothelial growth factor, and PLGF, placental growth factor, which is found in the blood. SFLT1 is overproduced by the placenta in patients with preeclampsia. When a patient is tested for preeclampsia biomarkers, an immunoassay using a sample of the patient's blood measures the soluble form of FLT1, which is again SFLT1, as well as PLGF, which is again placental growth factor, which is also a relevant biomarker for preeclampsia. If high levels of SFLT1 and low levels of PLGF are indicated, this suggests that the patient is at high risk for preeclampsia or has preeclampsia. The first thing we're going to do is type what we are looking for in the search bar. We are looking for FLT1. I like to give as much information as possible, so I'm going to include FMS, like tyrosine kinase 1. And little spelling error right there. Okay, hit search. Now I am looking for mRNA. So I'm going to click on gene. And I have some human, mouse, rat, but I want human. So I'm going to narrow my list down to human. Okay. And I have a few options here, but I want FLT1, human. So here we go right here, located on chromosome 13. All right, so you'll get some good information that you can use, what type of gene it is, protein coding. You can get a summary of it, and additional information. Now, again, I came here for mRNA, so I'm going to come over here and click on reference gene sequences. And we have isoform 2, isoform 3, isoform 4, isoform 1. Now, I know this looks out of order, but there really isn't necessarily a specific order. It's more so when this particular information was accepted by NCBI, when it was loaded by NCBI, is when its number was created, okay? But I am going with isoform 1 because that is the one found most in the population. Also, most likely, it is the one that was researched most. So I'm going to click on this NP number right here because I would like to show you something. And I'm going to click on FASTA. And you can see that we have MVSY. So this is not mRNA sequence. This is a protein sequence. This is methionine, valine, serine, tyrosine, etc., etc. Okay? So not quite what I want. So I'm going to go back. And I want isoform 1. And I'm going to click on the NM number. Again, FASTA. Some people call it FASTA as well. We know what you're talking about when you pronounce it, so pronounce it the way you pronounce it. All right, so here we go. All right, so this is our mRNA sequence. Now, we have adenine, thymine, cytosine, guanine. Now, I know I said this is mRNA, and I know that you are looking for uracil, but for GenBank, instead of using uracil, like you would expect, thymine is used. Okay, so it's just to help with comparison to DNA. That's the only reason why it's used. But you can see right here that this is the mRNA sequence. That is how easy it is for you to look up genetic sequences using GenBank.